Hey there, I'm Jen Cannell, and Researchers Under the Scope is back with a burning question, especially for anyone who's pregnant.
Meanwhile, marijuana use is becoming legal across the U.S., but how safe is marijuana use for pregnant people? A new study suggests it may affect a growing fetus as early as the first trimester.
A new study has linked marijuana use while pregnant to early childhood cancers. The study comes from Duke University's School of Medicine.
Tonight, more pregnant women are coming forward saying yes to pot. They're claiming it cures their morning sickness and anxiety.
It helped. My nausea, it went away completely, and I was actually able to eat.
A new study published in the Journal of the American Medical Association shows more pregnant women are choosing pot to treat morning sickness and anxiety.
Why, and this is a question, if somebody is pregnant, are they not aware that ingesting something like that could be dangerous to the fetus?
I think that we've had such a change in the United States with marijuana over the past 10 years with the legalization sweeping through that some people are viewing the risks of marijuana very differently now.
Marijuana and cannabis, what is the danger to a fetus? And how can we tell? Well, we'll put that question to Dr. John Howland. He's a behavioral neuroscientist studying cannabis in rats and mice, and he's our guest today on Researchers Under the Scope. Hey there, I'm so glad to have you back with us. We're recording this on Wednesday. It's the 18th of September, 2024. We're looking at pregnancy and cannabis with Dr. John Howland. He's a professor of anatomy, physiology, and pharmacology at the University of Saskatchewan's College of Medicine. And this spring, he and his team won a 5-year grant from the Canadian Institutes of Health Research. The idea is to study cannabis smoke exposure on pregnant rats and their offspring, just to see what that changes about their behavior, their neurophysiology, the way their brains are built, to see how it affects these baby rats and baby mice in their long-term development. Dr. Howland, welcome to the podcast.
Hi, Jen. It's great to be here. Thank you for the invitation.
Well, and we should also say congratulations, because that CIHR grant, that's worth just over $960,000. Way to go.
Yeah, it's pretty amazing. It was a bit of a journey to get it funded, as many grants are these days, but we were successful in the end. So here we are.
As we heard in that tape, things are kind of changing when it comes to cannabis, the way people see it, I think because it's been legalized in so many places. And we've had your co-principal investigator, Dr. Robert LaPrairie, on the podcast before talking about hotboxing rats and looking for the brain effects. Why is it that rats and mice kind of make the best model for this work?
Well, I think it's a combination of things. They're, similar enough to humans that we can gain some conclusions. They're also, have a short developmental time frame, so we could do the experiments quite quickly. And they're also affordable enough that we can actually generate enough data that we can make some conclusions that way. So it's sort of a balance of strengths and weaknesses.
And when you say they have a pretty short turnaround time, how long is that lifespan in terms of development?
Yeah, in our studies, pregnancy is about 3 weeks long, and then the offspring go through puberty around 40 days of age, and we would call them young adults sort of in the third and fourth postnatal month. So, in 6 months from receiving the to-be parents to testing the offspring's adult behavior. In a 5-year window, you can do a number of experiments and sort of refine your hypotheses and your thoughts as you go.
Do rats even like ingesting cannabis?
Yeah, this is an interesting question in the field, and I think the answer is under many circumstances, no. Lots of people use cannabis to relieve anxiety, but some people can feel more anxious after using cannabis, and there's evidence, certainly dose-dependently, that rodents also feel anxious when they ingest cannabis or are forced to ingest it. So, for instance, they won't really self-administer it that easily. There is a lab in Western Washington now that has developed a protocol to have rats, I guess it is all rats in their hands, choose to have a THC high cannabis vapor administered into a chamber. But for instance, in our model, we expose the animals and they breathe in the smoke of the combusted cannabis, but they don't have a choice in that regard. So.
And it's funny, in terms of your research career, if you think back to where this all began, did you ever think you'd be watching over a flock of mice or rats that occasionally have no choice but to go hotbox the place? Yeah,
it's an amazing change. I mean, I started doing this type of behavioral neuroscience work in the late 90s at U of S here in Saskatoon, and I just never considered that we would be where we are now. I mean, the legalization of recreational cannabis use in Canada has really changed the way that research can be conducted.
Did you always know that this is sort of the avenue you wanted to go down?
Yeah, I mean, that's interesting too. I mean, I started out here at Saskatoon as an undergraduate in psychology, actually, and I didn't really have a lot of clear direction, as many 18 or 19-year-olds may identify with. And I actually took a second-year physiological psychology course where we started talking much more about the brain mechanisms that mediate behavior. And I really just have never looked back from that. I got very excited by that course, and then I actually changed my degree path to become much more science-oriented. And I stayed in psychology when I went to Vancouver and did some graduate work at UBC. It just really was just trying to understand cognition and other good psychology processes like learning and memory or attention, and really how they are mediated by the brain and the mechanisms that underlie our behavior has fascinated me, well, for like 30 years. So yeah, it's pretty cool to end up in this place, and I'm pretty fortunate to be here.
And what part of the brain are you really going to be looking at? Like, is it the prefrontal cortex of these mice or rats?
Yeah, I mean, we call it cortical limbic circuits. So yes, the cortex is definitely involved, but we also look at other areas like the hippocampus and the amygdala. These are areas that are involved in higher level processes, like in the psychological types of processes. I think there's pretty good evidence that they are at least subtly affected after gestational exposure to cannabis. There's also an interesting area, my student will be excited if he listens to this, it's called the retrosplenial cortex. So we're going to look at it in quite a bit of detail too. It seems to be sort of involved in what's called this default mode network and sort of consciousness. So cortical areas for sure will be assessed in our experiments.
And going in terms of sort of the developmental origins of health in these rodents, what do we already know about pregnancy and infections, or if you're exposed to other toxins while you're pregnant?
Yeah, I mean, for sure. There's quite a long history now of research in this type of area. And I mean, I've actually had funding to study the infection type of story as well. And certainly mediators of these events that occur to the mother. So for infections, it could be inflammatory proteins. And for the cannabis exposure work we're doing now, it can obviously be the cannabis itself or the reaction the mother has to the cannabis. It does flow through the placenta and impact then the unborn offspring. And my particular work and interests revolve around how the developing brain is then altered as a result of that environmental exposure. I mean, there's really good evidence that other organs as well, like the heart would be an example, could be altered in utero, and that would set you then up, or metabolic programming, right? Risk of diabetes and stuff can be at least partially primed by events that happened in utero. So really that's something I've been studying since I started my own lab about 16, 17 years ago.
Yeah, I'm sort of wondering, like, what do we already see in terms of changes?
The most obvious example from the grant that was funded is that we do use strains that have high THC versus strains that have high CBD. Those two phytocannabinoids, or plant-produced cannabinoids, have the highest levels in the plant. And if you go to any cannabis retailer, you can find strains that differ on those two types of cannabinoids. So those cannabinoids, when ingested, affect distinct receptors in the brain and body of both the mother, the placenta, and the unborn fetus. So what we do know is that the endocannabinoid system, which is the cannabinoid system in our body.
Yeah, the onboard models.
Yeah. So we have our own molecules that stimulate these receptors, but when you ingest these plant-derived molecules, or you can ingest, artificial molecules, you may have heard of spice or different types of molecules that affect the cannabinoid system, we can then change the natural biology that occurs with the signaling at these receptors. And so these receptors are involved in many sort of discrete events during brain development. So like neurogenesis, or the creation of new neurons during brain development, where those neurons go, specifically how their processes are refined later. And this isn't exclusive to early development. This goes on, forever. Because we have the cannabinoid receptors throughout our lifetime. So that's why there's so much interest, for instance, in adolescent use as well, right? So we don't have any experiments planned right now that subject our rodents to cannabis in adolescence, but we are going to test other things in adolescence because that's a critical period of brain development. So really we're looking at brain development from its earliest origins in utero through early postnatal development, childhood, through adolescence, and then into sort of long-term health outcomes in adulthood.
And what's it going to look like for the little flock of mice that's going ahead and getting into the little smoke chamber that Dr. LaPrairie described? How long do you leave them in there, and what do you think you might start to see?
Yeah, so right now our model's pretty simple. We breed the animals and we know what day they got bred on. So then we start exposing them a few days after conception daily to a strain of cannabis, be it high-THC or high-CBD strain, and we expose them once per day for 15 exposures, which is about 2/3 of their pregnancy. And each exposure involves them breathing in the smoke of combusted cannabis for about 15 minutes. So it's like a daily exposure, and then that cannabis hangs around in the bloodstream for, several hours. We can do another episode some other time on sort of driving in cannabis and the questions around how long, something like THC stays in the blood. But certainly the animal's exposed for a period of hours, not minutes. And then we can look at levels in any tissue you can think of related to the mom, the placenta, or the fetus. Then the animals give birth naturally, and then they grow up. I mean, we've done tests as early as postnatal day 3, just looking at basic reflexes of the pups and that sort of thing. But really, the meat of the project is doing testing during adolescence and early adulthood. So we do a lot of behavioral testing in my lab. That's really what my specialty is. So looking at the function of these cortical limbic circuits I mentioned earlier. So we test memory, we test attention, we test sensory responsivity, that sort of thing. And then there's these other labs involved. We've mentioned Rob LaPrairie. His lab's involved in measuring the levels of these cannabinoids in the blood of the animals. And then there's a guy, Jeff Dong, who's actually also been on this podcast. He's an expert in understanding how gene expression might be altered in specific neurons. So we're really interested in how that exposure in utero months later might affect the expression of certain genes. There's pretty good evidence that some genes related to opiates and opiate use may be affected later in life in the amygdala. Some of that work's been done in New York in a lab run by a woman named Yasmin Hurd. But we're going to follow up some of that work from a genetic angle and look at some new brain structures, some of the ones I mentioned before. And then the third part is actually to really carefully assess how the brain's functioning in the offspring, and we're going to use some imaging approaches that allow us to monitor brain activity using these little fluorescent proteins. And this work is going to be done at the University of Alberta with one of my longtime collaborators, a guy named Ian Winship, and then another professor named Alan Chan. So they have these microscopes that allow you to measure patterns of activity over very large areas of the cortex, like pretty much the whole rodent cortex, or very micro structural changes that could occur at a very, very small, like a micron level of space in a neuron. So, the idea is to try to relate the behavioral changes I mentioned first with the types of exposures, then the changes in gene expression that could lead to changes really in circuit function. And there is some evidence from the human work that these types of circuits are altered. So for instance, you can do like fMRI, you get an idea of where blood flow is going in the brain in people. So there's been work like that done, and there have been changes seen in cortical networks. So we're going to try to look at those in the rodents. The big advantage here is we can really know exactly what our animals have been exposed to and what they've not been exposed to. So we can set up proper controls. If you start following people, they lead complicated lives, and there's a whole bunch of things going on. So really focusing that change in development or those changes in neural activity to cannabis is quite difficult. And we're hoping to be more controlled and more specific in the experiments we're going to conduct.
So down the road, where do you see this research being really useful? Where do you see it going?
Yeah, I think there's two ways that we talk about it. The first is simple, right? Public awareness of what cannabis does and knowledge. I'm always shocked when I talk to people about THC versus CBD, and they don't really understand the differences in those two molecules. And THC is obviously...
The one is that I'm going to be high, and the other is I'm going to be kind of relaxed and sleepy.
Yeah, and they have very different biology in how they work. So really filling out some knowledge in that regard, and we're going to try to communicate these findings so that people have more awareness. I always find it really interesting that, like the headlines you showed or we listened to at the start of this podcast, talk about legalization as being just this change and everyone thinks cannabis is safe now. But it turns out a lot of things that are legal, like alcohol and tobacco, aren't that safe during pregnancy either. So I think we just need more knowledge in this regard. And cannabis might be a little more complicated than alcohol, which has only real one ingredient that is causing the problems during pregnancy. Anyway, that's the first part. The second part is, if we understand how the brain is changed by exposure to cannabis, we may be able to develop therapeutics. And I don't necessarily mean big pharma and drugs. One of the experiments we've proposed in the grant is to look at how a different experience in adolescence might affect the long-term consequences of the exposure in utero. So for instance, if you were exposed in utero and then you had a series of stressful events in adolescence, the question is, does that exacerbate or make the phenotype or the behavioral change from the utero exposure worse? And the counter to that is if you do something that could be neuroprotective or beneficial. So what we've proposed is an exercise manipulation in adolescence. So if you were exposed in utero and then you have a more positive environment in adolescence, can you counteract some of the effects? So I think that could be quite interesting as people understand their exposures and the exposures of people in general. And the really good thing about something like exercise is the side effects are minimal. So engaging in that type of activity should be beneficial regardless. But is it even more beneficial for people who are exposed to cannabis and have these susceptibilities? These studies also will just really increase our understanding of how the endocannabinoid system leads to brain development. And one of the big, basic neuroscience questions is still how does the brain develop, and then how does it exist as this highly sophisticated, I hate to say supercomputer, but to use that cheesy analogy, this highly sophisticated, computer that can do all these things and I believe anyway govern at least a significant portion of our behavior as we grow in age. So, just understanding that's a critical question for modern biology. And the endocannabinoid system is certainly involved in that. So by manipulating it, we will gain information that way as well.
It's funny, as a person who's been pregnant before, I heard all the warnings, definitely stay away from alcohol, stay away from smoke, stay away from cannabis, deli meat, coffee, like you name it. We weren't supposed to have it. No sushi. And I get that. But especially in the tape that we were hearing, it sounds like a number of pregnant women have used at least some amount of cannabis. Human women have done it and gone, well, it's not like thalidomide. My kids aren't being born without limbs. But do you see in the future any point at which we can say to pregnant women, yes, if you have to, have a little for severe nausea, but don't do it too much, or don't do it too late or too early in this pregnancy?
Yeah, well, I think these are all questions that are there's a lot of gray here, right? You're right. It's definitely not like thalidomide. I mean, that's true. There's a few sort of from the epidemiological studies. Like, for instance, if you look at women who use cannabis during pregnancy, 0% initiate during pregnancy, right? Very, very few. And that's actually a problem with our model. And something we're going to try to change is we're going to expose the animals.
Your mama rats weren't stoners before.
No. So we are going to expose some animals before breeding. And you can also play, of course, the same game with the father. Yep. What happens if the father's a user? Obviously, nothing happens in utero, at least directly, but there are questions. So the majority of women, once they know they're pregnant, stop using. So there is more exposure during the early stages of pregnancy than the later stages. But to get to your nausea example, which is a really good one, I mean, there are medications that have been approved to treat nausea during pregnancy, and I guess I'm not an expert on really what the side effects or how strong the science is on using those medicines. But of course, as you just said, using anything during pregnancy is of question, at least. And I guess it would depend how severe the nausea is as well, right? I mean, if you literally can't eat and sleep, then the amount of stress you'll be under during your pregnancy will actually affect the fetus as well, right? I mean, it's very clear that stress during pregnancy can also have detrimental effects. So there's a balance here, and it's not black and white. And I mean, that's what I like. I like the complexity, but it's not simple. But I think more knowledge has to be helpful as women make these decisions for themselves.
Yeah, making a gray area just a little more clear. Yeah. John, thank you so much for being on the podcast today. This is great.
Yeah, it's wonderful. No, I hope we have some real world relevance here at the university.
You definitely do. So thank you.
Oh, yeah. Yeah, thank you.
Dr. John Howland is a behavioral neuroscientist who studies the factors that change our behavior, the way we remember things, the physiology of the brain. He also looks at diseases such as epilepsy and schizophrenia. John Howland's a professor of anatomy, physiology, and pharmacology at the University of Saskatchewan's College of Medicine. And if you'd like to see more of what his lab's working on, go to medicine.usask.ca. Researchers Under the Scope is a presentation of the Office of the Vice Dean of Research at the U of S College of Medicine. We recorded this podcast on Treaty 6 territory, and we pay our respect to the First Nations and Métis ancestors of this place. We'd like to reaffirm our relationship with one another. And this is the part where we say thank you for sharing your ears with us and tuning in. I'm Jen Cannell, I'm your host, and go ahead and share this episode with anyone who might have an interest in smoking weed from time to time, especially if they're in that neighborhood of life where pregnancy might be a thing. Go ahead and follow the show so you can stay up to date on Researchers Under the Scope.